Akshay. So I am going to use this talk which the Ashoka University outreach team helped put together. I mean, I kind of provided the content and they jazzed it up a little bit and stuff. And this is part of, this presentation is part of the talk that I gave us their outreach, as part of their outreach series called Scientifically Speaking. And given that it's going to do the same, it's going to have the same stuff that I'm going to be talking to you about, I figured I'll just use this as is. I'll today be talking to you about how we in the field think life chemically originated on the Earth. And prior to getting into some of the nitty-gritty, I will lay out a very broad context before we get into that. Yeah. So one of the important things to think about is how might have the fundamental individual pieces of life's grand puzzle, how did they come about? And when we think about these individual pieces, we immediately think about biomolecules, because if you sort of zoom into the most fundamental unit of all life, which is cells, you see that it is a bag of different molecules that are interacting with each other to essentially facilitate the various sort of events that allow us to sort of survive and have this conversation today, so on and so forth. Yeah. But if we also think about this biomolecule, one of the very crucial things that comes to mind is that there is this unbelievable continuum from physics to chemistry to biology. And what do I mean by that? Here on the bottom panel, you're looking at some of the monomers that make up the biomolecules. Here are amino acids, glycine, alanine, glutamic acid, and serine, which come together to make this protein here that you're seeing. It's a cytochrome C protein, and this protein goes back to some of the oldest living molecules on Earth, all the way to the base of the tree, close to the base of the tree of life. It's present in all three domains of life today, which is prokaryotes, eukaryotes, and archaea. And on the right-hand side here, you have an RNA molecule, which can fold back on itself and form this extremely beautiful engine called a ribozyme. It's called a ribozyme because it's a RNA enzyme, and it also can catalyze reactions just like your enzymes, proteinaceous enzymes can do, right? So if you zoom into these complex biomolecules, you can see that they are fundamentally built up by individual monomeric entities. You further zoom into them, you notice that they are, sorry?
Ma'am, the slides are not changing.
I haven't changed the slide yet. I'm moving my cursor. Oh, wait, you're not seeing a slide called Origin of Biomolecules?
No, we are seeing the title slide only.
Oh, crap. Man, this happens with Google Meet. We should have done this on Zoom, you know? It happened with one other outreach talk I gave a few months ago. This is screwed up. Okay, I'll have to get out of the... Oh, shit. Okay, hang on, please. This is terribly annoying. So let's do this. Okay, can you now see the full slide?
Yes, ma'am. Yeah.
I'm sorry. I will. If I go into slideshow mode, this is not going to happen. So I'll have to stick with this. Yeah.
Okay, ma'am.
Yeah. So let me just get a little bit of this going on the side. So, anyways, I was in the next slide. This slide where I was talking about the continuum. Can you see the slide now?
Yeah.
I'm very sorry. So the lower panel has your biomolecules. Here on the right side, bottommost region are the very complex biomolecules, one a protein-based molecule and one a nucleic acid biomolecule made of RNA in this case. And these are fundamentally made up of simpler monomers like glycine, glutamic acid, alanine, serine. These are all monomeric entities that make up proteins. You further zoom into them, you actually see that they are made up of even more simpler set of molecules that can form even more simpler entities which are only made up of a few different atoms. But they can build on, the few fundamental scaffold can actually build on itself and can make much, much more complex chemical molecules like buckyballs that's showed here. These buckyballs are found in the space. It's found in the interstellar medium, and this is a completely carbon-based molecule, and it can go up to C60 carbon atoms. You further zoom into them, you see that these molecules are made up of atoms. Yeah. So you see this continuum going all the way from physics Atoms interact to form molecules, simple molecules, chemistry. Simple molecules get together to form more complex molecules like fundamental monomers. And some of these monomers that I'm showing on this panel are relevant to extant biology or the biochemistry that we see on Earth today. So where do we think these molecules came about on the early Earth? Because we are talking about the transition from chemistry to biology to understand how life might have emerged on Earth. There are two ways that the field thinks about it. And sort of for those of you who are not aware, this broad area of research under which chemical origins of life comes is called astrobiology. The idea is that we are all made up of stardust. So what the molecules that start out in the nurseries where stars are made, we are made up of the same fundamental molecules, right? So within astrobiology, you have a whole bunch of fields of sciences that are crucial for understanding a lot of things, and we'll get to that as we go forward. But the idea is to try and understand eventually how did chemistry transition to biology, to understand the transition from non-life to life. So then the question is, where did the fundamental molecules come from? And the astrobiologists in the field think about it in two ways. One is exogenous delivery. What do we mean by exogenous delivery? A lot of the simpler molecules that made that kind of came together to make the more complex biomolecules might have just been delivered by comets, asteroids, and meteorites. And one other way to think about how these might have come on early Earth is to invoke syntheses on terrestrial environments. And here is an environment that is shown, which is called a terrestrial volcanic geothermal pool. It's terrestrial because it's on the surface of Earth. It's volcanic because it's a volcanic geothermal pool because it's right at the edge of a volcano. And there's a lot of geological activity and there's a lot of thermal fluxes that happens in these environments. And these kinds of environments, which are very rich in chemical and thermal fluxes, are very conducive to host and facilitate uphill reactions like polymerization. So if you want to go from monomers to polymers, you need to basically form these intermonomer linkages, whether it be a protein, it be a nucleic acid, or it be a carbohydrate, for example. You need to bring these monomeric entities. Let's think of a pearls on a string. The individual pearls are the monomers, and the string that holds them together is the intermonomer linkage. Any of these classes of biomolecules, proteins or nucleic acids or carbohydrates, involve an intermonomer linkage. In the case of proteins, it's called a peptide bond. In case of nucleic acids, it's called a phosphodiester bond. For it to come together and form these bonds is non-trivial because they all like to be single. They are not very happy mingling, right? In your cells and my cells, this mingling is forced by extremely precise molecular engines called proteins, and which are essentially, a lot of them are enzymes. But on early Earth, there is this whole chicken and egg paradox. You need the information coded in a nucleic acid to make a protein, but you need a protein to make that nucleic acid. So which came first? So I'll get to that conundrum in a second, but if you keep that conundrum aside, it is still important to understand what might have been the means by which these biomolecules would have come on Earth, and these are the two ways that they might have come on Earth. And I already gave this away, that the most fundamental unit of all life on Earth is cells. Whether it be Nelson Mandela here, T-Rex, your mushrooms, your giant sequoias, a supergiant catfish, or a microorganism. In this case, it's a bacteria. All of them, the one common thing that they have that binds them together is the fact that they're all made up of cells. So then, when you start asking the question, how did chemistry transition to biology? One of the easiest way to think about this question is to try and understand how did this fundamental unit of life come about from a bunch of interacting molecules. Here in the slide, I'm showing two kinds of cells. One is a prokaryotic cell, which is present in bacteria, and one is a eukaryotic cell, which is found in you and me and in other fungi and plants. The take-home message to show the slide to you all is to show that even the most simplest units of life, even the most fundamental unit of life, is very, very complex. Eukaryotic being more complex than prokaryotes, because in eukaryotes you have membrane-bound organelles, but in prokaryotes there are no real, very classic visual demarcations. Though the more we understand these systems, we're realizing that there is some very kind of beautiful way in which things still are kept separate by forming gradients, so on and so forth, but it's still very, very complex. So then the question is to ask, and one more thing, all these unicellular organisms, which are thought to have been one of the earliest forms of life that came about on Earth, have been able to colonize every single niche that you can think of on Earth, right? You have UV resistant organisms, you have heat loving organisms, hyperthermophiles or thermophiles, you have salt loving organisms, halophiles. This amount of salt will kill you and me. We cannot drink salt water, for the simple reason that our cells will not tolerate that amount of salt. Permafrost dwelling organisms that love low temperatures. So it's amazing how life is not just, even the most simplest forms of life on Earth today, or seemingly simplest forms of life today, have not just come about, but they have pretty much colonized all of the niches that are on Earth, which you and me will perceive as very extreme. Given this, one of the things I would like to put out there is that there is this notion of a tree of life. On the left side is a cartoon version of it. These are the 3 domains of life. Here you have the bacteria. Here you have the archaea. Way back, archaea was classified under bacteria. They were called, they have certain hallmarks which kind of are very different, so we have to put them in a separate domain. And there is this very famous theory called the endosymbiosis theory, which posits that a bacteria and archaea came together to form eukaryotes, which is you and me, right? Now, if you keep going back down, you see this thing here called LUCA. Can everybody see that? Yeah. I'm hoping yeah, okay, somebody's listening to me. Awesome. LUCA is the last universal common ancestor. And there is this notion, there has always been this notion when the tree of life was proposed, and even today, a lot of them think that this last universal common ancestor is the first complex being that came about, from which the 3 domains of life came about, right? But the more we understand biology and the biochemistry of how things function in our cells and how things might have happened, this is the understanding that is emerging, which is that it is probably not a single LUCA or a single kind of an entity, but it probably was a common ancestral community of primitive cells, which behaved a lot like multicellular entities, which are present even in modern era, but they would have been much, much more primitive. And what do I mean by primitive? Here is an artistic depiction of this most simplest of all cells that is thought to have emerged very early on, before the more complex bacteria came about, and these are called protocells. Protocells or primitive cells are literally entities where you have a lipid membrane. So imagine a tennis ball. The outer layer is a bilayered lipid membrane. And why do we think it's a lipid membrane? All of life, all the cells that you see in all the 3 domains of life end up using lipids as the outer covering. So there is probably a fair approximation when we say that maybe even the very primitive of cells did use some lipid-like molecules for the outer membrane, outer covering. And inside you have an encapsulated genetic material. Here you see this L-shaped structure that is an RNA ribosome molecule and some other related molecules that is inside the tennis ball. And what else is there, which we think now might be important, is minimal network of molecules which sort of interact to keep the cell alive, sustain it, and keep it keep it alive and allow it to grow, reproduce, and all that stuff. Sorry, this brings me to one very important thing. How do we define life? When I'm talking about emergence of life and origins of life and chemical origins of life, it is important to have a working definition of life. And if you actually go and say definition of life on Google, you're just going to get some crazy number of responses, right? Several hundreds of them. So it's just not, one, it has to be scientific. 2, it has to be something that we all in the field who come from very diverse areas agree upon on a minimalistic definition. So there was a committee that was ratified by the NASA Astrobiology Institute that came together and brainstormed, and about a couple of decades ago they came up with the definition for how the kind of definition of life that we work within, whose constraints we work. Life is a system of chemical entities that are capable of growth, replication, and molecular growth, replication, and evolution. If they have these hallmarks, and if the set of interacting chemical molecules can show these hallmarks, then one can probably consider them as being alive. And what we have been able to do thus far, and I have to point out that this is something where how did life come about? How did we come about? These are questions that are not being asked only today. These are questions that have been asked for time immemorial, right? But
Yeah.
the more scientific sort of way, the more scientific way of approaching this question gained foothold more in the early 20th century. And it started off with this beautiful theory that was put forth by two people, and I'll get to that in a second. But the field of prebiotic chemistry, which is the chemistry that deals with the chemistry of life, was established as an area of science only in the 1950s. But since then, we have work in this prebiotic chemistry area and in astrobiology at large has allowed us to come about with this wonderful story, which is consistent with evidence. In science, you need evidence, right? With experiments in chemistry, with what we know about the geology of Earth, the settings and what allowed for our planet to have a life, and with what biology has been revealing about some of the oldest forms of life. Some of the oldest forms go back, I mean, right now the record is about 3.85 billion years ago. These the cyanobacteria kind of cells were present on Earth, right? And after this whole time from 1953, we are now at a place where some of the pieces of this puzzle is coming into view, and it's telling us a lot about how this transition might have happened. I'm moving to the slide. So astrobiology, as I already alluded to, feeds off from various areas of science. It's heavily interdisciplinary. I am a biochemist by training. I did my PhD on sickle cell anemia. If anybody of you out here is interested, I did not work in the origins of life field at all. I worked on sickle cell anemia. I was looking at protein aggregation disorders at large. I was very fascinated with Alzheimer's and Parkinson's. I went on to do my postdoc in Parkinson's, but a very interesting set of events, which was completely unplanned, and hence I call it cosmic convergence of events, right? Happened, and then I stumbled into this field completely by chance in 2004. I completely fell in love with this field. I had a beautiful mentor who just was mind-blowing, and before I knew, I was like, I wanted to be a part of this. And I back in my postdoc and more recently we in the lab, tend to use tools from biochemistry, molecular biology, biophysics, organic chemistry, inorganic chemistry, and geochemistry to ask a lot of the questions that we are interested in. And also, somebody's child is saying something. Maybe you want to cut your audio off. And we also collaborate with people who are not experimental people at all, but they use modeling and theory as a way to understand these questions too. So when multiple minds come together, then the chances of finding out something super cool is much larger. So this area, I think for that reason, is absolutely attractive to me. It's extremely interdisciplinary, right? Another colleague of ours in ECS, she's a person whose work has implications for understanding some of the oldest fossil evidences that might be there. And this is Devapriya, she's an ECS, and she's a paleontologist. So we can use these multiple tools, and in our lab, I'm sorry, and hopefully in my lifetime, we'll try and figure out how did we go from these fundamental, simple biological monomers or complex chemical entities to the protocells, the oldest of primitive cells, which then eventually gave way to life as we understand it today. When did all of these events happen, or when do we think all of these events happen? And this is based on evidence, and I'm just not giving a hand-wavy time. It is thought to have happened about 3.8 to 4.2 billion years ago. Very early on in life, there was a very interesting bunch of reactions happening that set the stage for the emergence of monomers that's relevant to what we see in extant biochemistry or modern biochemistry today. And from that space, it's thought that molecules of an RNA world emerged, which probably set the stage for transition of this chemistry to biology. And what is an RNA world? RNA world is posited. RNA world hypothesis posits that very early on in the early history of life on Earth, the most primitive of life forms used just one biomolecule to do all the important functions of life. And what are the important functions of life, which I alluded to in the definition? You need to be able to grow. Yeah, so metabolism, RNA is capable, RNA enzymes are capable of metabolic activities. You need to be able to replicate, which means you need to be able to encode data, which can then be used. The information in that molecule can be used to make new molecules. RNA can do that, just like DNA. And RNA also can facilitate catalysis, like in metabolic reactions. So instead of thinking about how multiple molecules came about from the very complex prebiotic soup, if we can even try and understand how RNA did come about from the earlier, that will sort of reduce the complexity of the problem that we have to work with. Because modern biochemistry uses multiple classes of biomolecules. DNA and RNA encode information and RNA processes information. DNA's sole job is encoding information. RNA's job is encoding information and processing information, because even to date, some entities use RNA as the information molecule. For example, your SARS-CoV-2 virus. Whether it's alive or not is a different question, and according to the definition that I gave you, we don't consider viruses living. But there are a small group of people in the field who will vehemently say there's a fourth domain of life, and they call it, I don't know, viria or something like that, right? So even to date, there are entities which encode information in RNA. So DNA, RNA encode and RNA processes information, and proteins completely facilitate the functioning of the cells. But very early on, RNA could have done all of these things, maybe not as effectively, but it still could do it. So that all we then have to think about is, how did you go from the prebiotic chemistry space to a minimal world space where the molecules of an RNA world came about? How did they come about? And this clock here is sort of a way to think about this early history of life on Earth. Oldest known rocks which harbor signs of life go back all the way to around 3.85 to 4 billion years ago, right? But prior to that, what happened? You first had to form the solar system. It's thought to have happened about 4.5 billion years ago. And this formation of the moon, there are probably physics people here in this crowd, so I'm not going to talk about this too much, and they might be able to answer some of your questions because I know limited stuff about this, but it is thought that, and it's still debated, it is thought that these two bodies, a Mars-sized body called Theia, is thought to have crashed into Earth when Earth was still in a very molten state, and their contents got all mushed up, and then a small body got ejected, which got trapped within the gravitation of the newly formed Earth, and that is basically Earth's satellite, the moon. Now, this is a very important aspect pertaining to early Earth, which is thought to have happened around 3.8 to 4.1 billion years ago. Again, if you talk to some astronomers, there is a difference of opinion, but most of them seem to agree that there was a period around 3.8 to 4.1 billion years ago where there was a lot of bombardment of meteorites and comets onto Earth. How do we know this? Look at the moon. There's a lot of these craters, and that is also a proof that this was something that happened. And why is this interesting in the origins of life context? Many of these impact events are thought to have brought in the fundamental molecules that set the stage for the transition from simple chemistry to complex chemistry on Earth. And subsequent to that, around, yeah, so this is just to give you a sense of appreciation of how Earth is thought to have been around 3.8 to 4 billion years ago. Looks ultra hostile, right? But even during that time, there was a very interesting set of chemistry happening, and there's a lot of very interesting set of simple biochemistry that was happening that led to the emergence of protocells and some of the earliest forms of life. How does Earth look today? The famous phrase, right? A pale blue dot, like Carl Sagan said. It just looks like a stunning marble, no? But the common thing to both these kinds of Earths, the one that existed 3.8 to 4.1 billion years ago and the one you see today, they harbored these volcanic activities on there. I mean, volcanoes existed even back then. And these volcanic geothermal pools, which you can see even today on the right side panel, you see this beautiful blue, green, yellow sort of pool that exists even today. And very early on, very early in history, Earth's history, volcanoes existed. And these geothermal pools are thought to have been extremely important in the context of what might have been the earliest niches on Earth that might have supported some of these reactions I will touch upon in a second. We'll leave the poll here. These are the environments, right? Depending on whom you talk to in the field, there are two major contenders. One are the hydrothermal vents, which are on the bottom of the oceans. Again, these are also a consequence of geological activity. These are because of the smoke that you see coming out is because of volcanoes that is happening in the ocean floor. And here are the terrestrial geothermal pools, which are also showing these spewing smoke-like thing, right? Even this is a consequence of volcanic geological activity that is happening on Earth. And what is very interesting is, if you want to have a very unbiased approach, there are certain reactions pertinent to understanding some of the transitions that we're interested in that are very happily facilitated in these hydrothermal vents, and a good bit of them that are facilitated in the geothermal pools. So the jury is still out there as to where life originated. Having said that, in our lab, we do experiments in this context where we try and simulate volcanic geothermal conditions to try and understand some of the questions we're interested in. And these are pictures of certain other hot springs. I unfortunately did not find extremely cool looking hot spring pictures from India, but there are hot springs in India, and we actually have started to wear the hats of field biologists or field experimentalists by going out to some of these hot springs. We have not done that too often, but we did this once roughly 4 years ago and came back with samples to work with in the lab. We did some experiments there too. But this is the model that we work with. The idea is that if you have this tidal pool at the edge of a terrestrial hot spring, or you have the terrestrial hot spring harboring some of the fundamental molecules that are pertinent to the emergence of a protocell, geological cycles that are naturally supported in these environments would have facilitated multiple wet-dry cycles. Even you see them happening today. Back then, it would have been, even now you see it happening in the same fashion. It can be driven by day-night cycles. It can be driven by seasonal changes. Yeah. So if you had, let's say, the monomer molecules that make up the membrane, and if you have the monomer molecules that make up the genetic RNA, genetic molecule or the RNA, during a dehydration phase, your lipid molecules, which can be present in multiple complex organized structures. You have a micelle here, you have a bilayer here, you have a vesicle here, like a bilayered structure, but it folds back on itself and forms a tennis ball. Any of these entities can actually form these multilamellar sandwiches. And if you have the right kind of solutes present in this pool, they can get constrained now within a two-dimensional space. What happens in a three-dimensional space is the chances for interaction is much less. And I already told you about this thermodynamic constraint where these monomeric molecules like to be single. They don't like to mingle. So now you have to force them to come together to form the polymers. So this natural wet-dry cycle can facilitate that. So during the dehydration phase, you can actually entrap these molecules now into a two-dimensional space where they're now in closer proximity and they're moving only in 2 dimensions. And then the chances of collisions between the reactive groups is much higher. Here is an inset of these dehydrated lipid matrices. It's a freeze fracture image from using microscopy as a technique. Unfortunately, you can't see it very well. But the idea is that on a subsequent rehydration step, you can actually start now having entities emerge that look a lot like the protocell I was talking to you about now, because the dehydration phase is going to now allow the formation of polymers, informational polymers, and a subsequent rehydration phase can now allow for the encapsulation of these informational polymers into a membranous vesicle, very similar to the protocell that I showed you. And selection. How do we drive selection here? We don't have to do anything, just the physicochemical parameters that existed then, your pH, your temperature, the ions present in the environment. A lot of ions will depend upon the rock aqueous chemistry, right, of the volcanic geothermal pool. All of these things can act as selection pressures, and it would have probably selected for a subset of protocells, which then can undergo this cycle over and over again. And then you can imagine some form of primitive evolution even in this space. So with this thing in the background, we kind of understand some of the things we're interested in. But like I said, the stage was set by these two amazing scientists. Here is Operin and here is Haldane. Operin, a Russian scientist, Haldane, an American scientist who had an Indian connection, very fascinating Indian connection. The two of them independently proposed this hypothesis where they said that on the early Earth, in the primitive ocean on the early Earth, where you had the right fundamental inorganic ingredients like methylene, dicarbon, water. Water is the canvas in which all of this is happening, right? Ammonia, carbon dioxide. If you had these molecules present in the primitive ocean with the right kind, with some very good energy source like lightning in the early atmosphere or ultraviolet radiation that comes from sun, and early on in during that period in time, the ultraviolet radiation is thought to have been intense, you can actually have very interesting reactions where you can go from inorganic molecules to organic molecules like the kinds we see which make up your proteins. For example, some of, this is what they proposed. They said that early Earth was very reducing, so rich in electrons, which facilitated these reactions wherein you can go from an inorganic molecular space to an organic molecular space. But nothing in science is fully, one can't make a total claim till one can systematically demonstrate it in an experiment, right? And this is where Stanley Miller and Harold Urey come into picture. Harold Urey, I think, was giving a talk in Chicago. I might have the city wrong, but I think it was in University of Chicago. He was giving a talk and he was talking about this O'Brien-Haldane hypothesis and generally speculating about this. And Stanley Miller sort of, after the talk, went up to him and said, even I've been thinking a lot about this and I have ideas about how I would like to demonstrate this using an experiment. And before you know, Miller went to Urey's lab as a postdoc and went on to, for the first time, show that it's not just a hypothesis. One can actually demonstrate this, start in this prebiotic simulation setup, right? This is the Urey-Miller apparatus. This is it's called the Miller apparatus. Here is the prebiotic ocean, and in the prebiotic ocean, you had all the starting reactants: water, dihydrogen, ammonia, methane, and it was very hot on earlier, depending on whom you talk to. The temperatures can range anywhere from 60 to 110, 120. But suffice to say, a good consensus is 80, 90 was very possible. Things start cooking, and then you have evaporation. And once these molecules came in the vapor state to this space, you had these tungsten electrodes giving out a huge amount of electricity, and a lot of interesting things happened here. And then it came to the space where you had, it's a condenser, you can actually cool down whatever's here, it's like super high temperatures, right? You can cool it down and then you can basically keep doing this cycle. Miller did something very amazing. He did this super systematically. And he went on to show that from this bunch of simple inorganic molecules, you can actually make hydroxy acids and amino acids. And back then in 1953, what was the kind of technique that you had to show the formation of these molecules? I guess a lot of you might have heard about it. It's called paper chromatography. Yeah, literally using a paper coated with silica to show that molecules can separate from each other based on a lot of different properties. Their charge, you can separate them based on size, so on and so forth. And that was the technique they used to show that these things are forming. A little trivia here. I know the second last student of Miller, him and the first grad student of Miller, Jeff Bada, Jim Cleaves and Jeff Bada got together. Stanley Miller was such an amazing scientist and almost anal, I would say, in the sense that he systematically saved some of the samples from back then and had extremely rigorous notes. And it was so straightforward for Jeff and Jim to go back and retrieve those experiments. And about, I want to say 14 years ago, maybe 10 plus years ago, they once again analyzed the products of these reactions using some of the very high-end analytical techniques available today. One of it is, it's called Icp Ms. And they went on to show that you not only form some of the simpler hydroxy acids and amino acids, but you also form the more complex amino acids in few parts per billion amounts, including phenylalanine, tryptophan, tyrosine, your ringed amino acids. And they went on to also replicate the experiment in a recently concocted Miller experimental setup. And they went on to show that, yes, indeed, you can go from the starting ingredients to all these interesting organic molecules. And after all these decades of work from 53 onwards, the field is at a stage now where we have been able to figure out that from the same set of starting ingredients, you can make the precursors you can make amino acid precursors, which essentially will lead to the class of biomolecules called proteins. You can make the fundamental precursors that make up your DNA and RNA. In this case, it's nucleosides, which is your nitrogenous base and the sugar minus the phosphate. And you can also make lipid precursors, which go on to make your membranes. So this is where we are now in terms of understanding of prebiotic chemistry. One of the big things that still is lacking is how do you go from these monomeric entities to polymers? That is still something that we do not systematically understand. And what might have these earliest forms of life look like? Again, going back to that protocell image, right? So this is an artistic depiction of it, but we work with protocells in the lab. We vary the membrane composition to try and understand how they respond to different physicochemical parameters that might have been present on the early Earth. We try to understand the makeup of the membrane on its own. We try and understand how the polymer inside the genetic polymer, how was that formed in isolation? Because eventually the holy grail is to bring the two of them together and see if they can become alive, which is grow, replicate, and evolve. So then you would get to this common ancestral community of cells from which you started forming the more complex single cellular entities, which is thought to have populated the Earth almost 3.5 billion years ago. Here is an image of gliocaspal cells. These are essentially cyanobacteria. And these are thought to have come about. These are the oldest geological evidences that exist on Earth. Here on the top panel in this central image, you're actually seeing these stone pillow-like structures. These are called stromatolites. Stromatolites are a product of interaction between biology and geology. It's essentially these microorganisms trapped in sediments, which just got layered and layered and layered and layered and formed these pillow-like structures. So take one of these stone pillows and cut them, and this is the section you're going to see. You're going to see alternating layers of geology and biology, and people have used radiocarbon dating to try and understand how old these molecules are that are present from the biological area, from the biological sort of slice, right? One of the cool things that happen is all this drama that I've been talking about thus far is thought to have happened when Earth was extremely anoxic, no oxygen. Yeah, right now you have some X% of oxygen on Earth, right? Even then, the most prominent gas on Earth is what? Is it oxygen? No? Yes?
Nitrogen.
No?
Nitrogen.
No, nitrogen.
Nitrogen. But you also have oxygen to some extent now, which allows us to be alive. But prior to 3.5 or 4 billion years ago, there was no oxygen. It was all under anoxic conditions. It is these amazing single molecular entities called cyanobacteria that is thought to have oxygenated the Earth to the extent that we see today. What did they do? They use solar energy, they use water, they use carbon dioxide, to fuel the metabolism or the fundamental reactions that allow it to produce the food that it requires. And oxygen was a byproduct, which then got shoved out into the environment. All the oxygen that you see on Earth today, the oxygenation event that led to it is thought to have been predominantly taken care of by cyanobacteria. Only 2 billion years ago, you had the eukaryotes emerge. About roughly 1.5 billion or 2 billion years after the prokaryotes emerged. These are more complex cells which have membrane-bound organelles, and that allows them to do a lot of other interesting things and have a lot of crosstalk between each other. And then here you see a choanoflagellate rosette colony where multiple cells are talking to each other. And at some point in time, there was emergence of multicellularity, and then we come to the space around 540 to 590 million years ago, which is called the Ediacaran period, where some of the most fascinating complex creatures are thought to have come about. And here, between 480 540 million years ago, you had this Cambrian explosion, which is thought to have led to a huge emergence of huge menagerie of creatures which was never seen before. How do we know about all of these guys? If you did not have bony structures in the organisms, you cannot have fossils. But once the bony skeletons came about, you could actually have these things leave imprints, right? Or bone chitin-like structures. So you could actually study them systematically using this field of science called paleontology to understand how old these are and try and date them. And then came this famous period, which we all are aware of, right? This is thought to have happened about 65 to 200 million years ago. In that 2.5 billion years ago is when you went from single cellular entities to all the way to dinosaurs. I mean, you had the simplest of eukaryotes form and then very quickly evolved into all the way to, I mean, you also had mammals along with the dinosaurs. Only thing is, when this Cretaceous tertiary mass extinction event happened, which is thought to have been triggered by asteroid strike in the eastern part of Mexico state, that along with a lot of intense volcanic activity right under where we are sitting in the Deccan Plateau is thought to have set the stage for events that essentially wiped the dinosaurs out, which allowed the more simpler mammals, a mole-like entity present back then. I don't know if I have that image. I do, here. This is supposed to be our ancestor, this mole-like entity, right? This mammal is thought to be our ancestor, like super ancestor, from which all of the other mammals came about, evolved and came about and stuff. When did we come about? On this clock, right? We literally appeared a minute or two before midnight. So all that happened prior to our appearance is extremely crucial for letting us emerge in the first place. So I think it's super important for us to not forget that intelligent, evolved entities cannot happen till more simpler entities would have first happened from even more simpler inorganic, and I mean simpler organic and further, if you go further back in time, inorganic entities. So super crucial to remember that you don't go directly from nothing to humans. There was all this grand drama that happened prior to our appearance. And in our lab, what do we do? Just to give you a sense, we try and understand how these primitive cells or protocells came about and what are the events prior to their emergence that would have played an important role for them to come about in the first place. So we work in the realm of trying to understand how the fundamental building blocks formed to make these genetic molecules. We're very interested in understanding how they come together to form polymers in the first place that can encode information. And like I said, there's this chicken and egg problem, right? So if you keep the problem of proteins aside, RNA alone can take care of both encoding information and catalyzing reactions, reducing our headache to try and understand came about and how did they replicate information as faithfully as possible, even in the absence of enzymes. And you will be surprised to know that just chemistry-driven replication of information is really not that crappy at all, as I had imagined when I didn't work in this area. And you know, during all of these events, in the backdrop, you had formation of and evolution of complexity in the primitive membranes, which essentially form the outer compartment. And then eventually these two entities come together along with encapsulation of a minimal network, metabolic network that can form energy to lead to the formation of these protocellular entities. We are interested in a few different projects. I can talk about it in detail in questions Q&A session, but the idea is we are also very interested in understanding the diversity in this RNA world space. RNA molecule, like I said, encodes information just like DNA, but it does it a little more shittily. That is because RNA is much more prone to mutation. But very early on, that might have not been a bad thing, right? Because if you want to sample all the sequence space of all possible, say, 50 mer RNA molecules, it is 4 raised to 50 possibilities. 4 raised to 50 possibilities. Even 4 raised to 21 possibilities is like 10 raised to 15 molecules. It's a mind-boggling number. So if one has to think of how quickly can evolution sample all of the sequence space to pick out the most interesting functional molecules, it's okay for the molecule to be a little crappy, because then that way you can sample the sequence space in a much smaller time scale, say in millions of years rather than billions of years, right? Or several hundred thousands of years. So we are interested in understanding aspects about how the prebiotic pool of nucleotides led to the formation of polymers, no enzymes again, hence non-enzymatic, and how once these polymers come about, how do they replicate information, again in the absence of enzymes, what kind of compositional diversity existed in this RNA world space, which eventually allowed in the emergence of the first enzymes on Earth, which are RNA enzymes or ribozymes. Again, we are also interested a lot in the external compartment. This landscape of primitive membranes, what it might have looked like, what are the different kinds of amphiphilic entities might have even been present that could have been incorporated in this membranous outer layer is something that we have very little information about. And there are a couple of projects in the lab where we are very interested in trying to understand these things. We are very curious also to know how might, once you kind of narrow down on a set of different amphiphiles and their derivatives, we are very interested in understanding how you go from, say, something very primitive in terms of compartments. And when I keep saying membrane compartments and lipids, our modern membranes have something called phospholipids, which is essentially one head and two tails. But very early on in prebiotic soup, it's thought that even to have this simple monomer of phospholipid emerge, it might have been non-trivial. So fatty acids are thought to have been what might have been used in the primitive compartments, because fatty acids is one head and one tail. They are thought to have been either delivered by meteorites, because if you actually, there was a meteorite that fell in Australia, in Victoria, in the 60s, Murchison meteorite. If you drill to the core of it, and people have done this, they drilled to the core of it and extracted the organic matter, and they saw that they could readily have vesicular structures emerge from them. And subsequently, they used high-end instrumentation to show that you can have about C6 to C12 hydrocarbons present in these organic mixtures extracted from the core of the meteorites. And yes, this is exogenous delivery. Then can we have terrestrial reactions that can form fatty acids? Yes, you have Fischer-Tropsch-like synthesis that can happen when you have when you basically have water and carbon monoxide, high temperature, high pressure. You can form beautiful hydrocarbons, which can then just have an acid group attached onto the head to form fatty acids. And then you just don't have fatty acids, but you can have the head group vary, and you can have a lot of different kinds of head groups, and hence you can have different derivatives of fatty acids that could have potentially been present on earlier just from terrestrial reactions that would have happened. We are also interested in understanding how minimal amino acid-based enzymes came about. Very broadly speaking, we're interested in evolution of very simple biocatalysts. And like I said, about 4 years ago, we started to even do some field-based experiments. That was the first time we went out, so we were super rusty, we were super amateurs, right? Here are the more hardcore geochemists out there. This is Chaitanya Mungi here, for some of you who might recognize him. He works in the Science Activity Center. He was my first grad student. Chaitanya, Nirja, my second grad student, and I went out to Ladakh, and we actually sampled some of the hot spring water samples, brought it back to lab, and tried to understand some of these reactions which we're interested in. How might they pan out under real analog conditions? Because in the lab, what are you doing is you're trying to oversimplify your problem. You're using a very reductionist approach to understand the fundamental mechanisms of how things might happen. And it is an important thing to do. But once we have come to a place where some of this fundamental understanding has happened, it's extremely important to try and now say, okay, analog conditions will the experiments happen in the same fashion? Because here you have a lot of other chemistry happening, and we don't even account for that in our reactions. For example, if you take this polymerization of monomers, right? In the lab, we do it in buffers. We simulate these wet-dry cycles, which would have been present on early Earth. We simulate either acidic geothermal pools or alkaline geothermal pools and try to understand some of these things. But again, it's happening only in a very sort of minimalistic scenario where maybe 1 or 2 ions might be in the picture. But then if you bring back samples from these areas, you actually see that you can have various ions that are released because of this aqueous chemistry that happens with the rocks. Then the question is, the processes that you're trying to understand, do they pan out in the same way, or is something else happening? We have no clue, and our lab is one of the few labs in the world that tries to understand this at great detail. We call it the co-solute problem, or the co-solute set of reactions, right? So at the end of it, I'd like to say that the origins problem could be very naughty, may seem very naughty, but it is not insoluble, right? Or insolvable. Eventually, since 1953, there's enough things happen that has already set the stage for a lot of the understanding that we have today, from where we sort of take off and try to understand slightly more complex problems. So yeah, naughty but not insolvable, and happy to talk about things that you have, questions that you might have. And again, like I said, we don't exist in isolation at all. Yes, astrobiology is a very less represented area of research in India. It is up and coming, and I'm happy to share that a few of us have come together to form this society, and we're trying to talk to ISRO to try and get them to involve us more in some of their missions. And now with this whole plan that ISRO has about putting humans in space, we're getting more traction from ISRO. Otherwise, ISRO didn't take me seriously say in 2013, right? They were like, yeah, astrobiologist, very nice, welcome back to India, but I don't know what to do with you, because they like people who do technology business. But now they're realizing the super cool implications that the fundamental research of trying to understand how life emerged has for understanding if there is life elsewhere in any of the exoplanets, and if yes, how we might even look for them, right? This whole area of research is now called habitability. And some of the physics people in the house today, or who are here today, might understand this or might have heard about this. But like you see, we have interactions with people in chemistry, in ECS. Gyana from ECS is a very important collaborator for us. He helps us understand the geochemistry, which sometimes flies over our head, right? But I think if you want to be in this area of research, if this interests you, the only fundamental personality trait that you need is to be extremely open. Yes, you need to train yourself in one favorite area, so to speak, or sort of narrow down your interest to some. I like to be at the interface of chemistry, biology, and say a little bit of geochemistry. Maybe somebody else likes to do it in the realm of physics and biology, and besides, say, astronomy and chemistry. I mean, I have friends who are astrochemists who try and understand the chemistry in the interstellar medium, right? Or try and understand the chemistry on your meteorites. But yeah, if you have an open mind and if you're open to collaborations, astrobiology is a fun place to be, fun science to be doing. Thank you for your time. Happy to take some questions. Like I said, I've kept it extremely broad, but we can talk about more specific things if you want to say something. I'm going to stop sharing now. Is that okay?
Yeah, ma'am. Thank you.
Hi, Sudha. This is
Hi.
Netra.
Hi, Netra.
Hi. So I have one question regarding when you were talking about the wet-dry cycles. I understood why dry cycles or dehydration would facilitate polymers.
Yeah.
But how would wet cycles facilitate incorporation into lipids?
So sorry, I don't delve upon it too much. Here's the thing. So you when you have this dry cycle, let me just try and pull up that. Give me 1 second. Na?
I think it was the 19th slide.
19th slide, not 19th, even further. No, 18th. You were right. 17th. Yeah. So let me present now. A window and share. So you very correctly pointed out that the dehydration phase readily allows for the formation of polymers, which are uphill reactions where you have to knock a water molecule off. So all these polymerization reactions, which happen even in your cells and my cells, are fundamentally condensation reactions. When I say condensation reactions, it means you knock a water molecule off, right? And this is done in our cells using high-end enzymes. But very early on Earth, no enzymes. How do you do it? Just let the natural geological events of wet-dry cycles facilitate this. During the dehydration phase, you have loss of water, so reduced water activity, so condensation is feasible. Now, what happens during the rehydration phases, you see these dehydrated matrices, the parallel stacks. These are lipidic, white strands are the lipid stacks. Now, what they end up doing is, you see this bottom panel here of these blue heads and turquoise heads with black tails. This is essentially a cartoon of a bilayer. And the water-loving groups are on the outside and the water-hating black tails are on the inside. And here is the chemical version of these fatty acids, right? Oops, what happened? Remove. Can you see my presentation still?
Yes.
I can't see. You're presenting to everyone. Thank you very much, but I need to see my screen now. Hang on 1 second, something happened. Some extension business happened. So you can now you can imagine this much more readily, because then water activity is going less, molecules are coming closer, and you can facilitate these uphill reactions. What happens in a rehydration phase is water is coming, a lot of water is happening. These water-hating groups absolutely do not like interacting with water. So they will immediately sort of push out the water. This is driven by an entropic effect. It pushes out the water and then it balls up to form again these vesicular structures. And when it does that, these abhi jahan pe yahan pe individual dots dikh rahe hain, here you're seeing kind of short red strings. These are representative of the growing informational polymers. Now they can randomly get incorporated into a subset of these blebbing out vesicles, right? So that is how they get incorporated. It is a completely random process, and one level of selection happens right there. Some vesicles might randomly have these polymers, some vesicles might not. Some vesicles might have polymers that can possibly protect this outside membrane by providing them some form of stability with their interaction. So that can also be envisaged as a kind of selection that might have come into play. And over multiple rounds of these wet-dry cycles, you can eventually have those protocells emerge, which now have interesting genetic molecules entrapped in them. Initially, it happened completely randomly, but with time, there is some sort of a crosstalk between them that selects for the more robust ones, which have more interesting molecules entrapped. Does that make sense?
Yes, it makes sense. Yeah, it makes sense. Thank you.
You're welcome. I'll stay with the slides just in case somebody has some other question.
Hello, ma'am.
Yama?
Ma'am, I understand that.
Can you introduce yourself, please?
Yeah, ma'am. I'm Aditya. I'm a third year BSMS student.
Aditya, tell me.
Yes, ma'am. So, like, the Harold-Urey experiment kind of showed that we were able to produce small molecules, right?
Right.
Small, simple molecules from our prebiotic soup, kind of.
Yep.
So, but I didn't understand, like, why certain molecules are more ubiquitous in biological systems than others.
Very interesting question, right? So a lot of these events that I've been talking about happened 4 billion years ago, 3.89 billion years ago, right?
Right, yeah.
Primitive life might have used a whole array of interesting molecules, which remained interesting only in those specific contexts. For example, I will use an example that will tangentially hopefully answer your question, right? If you take RNA monomers, which is adenine containing RNA, fundamental monomer, adenine, guanine, thymine, or uracil or cytosine, you will say, Sudha, all you have to do is knock water molecules off to make a phosphodiester linkage. So let's take these guys and cook them together. Okay, let's take them and cook them together, knock water off, right?
Right.
What happens is you end up forming very short dimers. It happens, but beyond that you don't, because the molecules are frozen. The monomers are frozen in a disordered crystalline matrix when you're just starting out with water and the monomers. But now you start off with these vesicles. You also have these monomers in the vicinity, and then you do a wet-dry cycle. You now start seeing small strings of oligomers, 2-mer, 3-mer, so on and so forth, 4-mer. The problem is the following. The backbone, the phosphodiester linkage, happily forms only under acidic conditions. You need to make the OH on the phosphate become a good leaving group. It's not a good leaving group. You have to make it an OH2. You have to protonate it. For doing that, you have to bring the pH down. Voila, phosphodiester happens. But the problem is at that pH of 2 or 2.5, your glycosidic linkage, which attaches your adenine, your guanine, your uracil, your cytosine to the ribose,
start breaking up.
kaput, gone. So then am I making informational polymers of an RNA world? The information storage happens in the base, which essentially can replicate because of hydrogen bonding with complementary bases in the other strand, right?
Right.
The informational molecules, hey, he nahi. So one of the important things I have to point out because of your fantastic question is that we in the field now, a good number of us are of the consensus that you can't directly go from a prebiotic soup space to an RNA world space. And there's a greater consensus that prior to the RNA world, you probably had you had pre-RNA and proto-RNA worlds where you had informational molecules that used a whole bunch of different bases.
Okay.
Which are not AU, GC, and T, because they can't stay stuck on your ribose. But if you veer away from your AU, GC, Ts, right, and you move to another whole space, let me stop. Let's go to the slide. Now, you see what I'm saying? You see these sugar phosphate backbones?
Right, yeah.
You see them? That are much more readily, that can super readily form under prebiotically relevant conditions, acidic geothermal pools. You take ribose attached to a phosphate, that monomer is called a ribose monophosphate, and you put them through wet-dry cycles, they beautifully form these oligomers.
Right, yes.
Then the question is, can we now try and sample a base space which is very different from our modern bases, AUGCT, and see if any of them can condense onto this oligomer to form pre-RNA world polymers. I'm happy to share that you can form a pre-RNA world monomer, and that monomer can oligomerize, but we are still struggling with forming an oligomeric backbone onto which we can systematically show that you can have these other kinds of bases kind of condense on and form this beautiful informational polymer now, which doesn't look anything like your DNA or RNA, but looks very similar to it. Looks like its cousin. Why am I saying all of this?
The question then arises is, how can you guess that they're such molecules? Like you don't have any kind.
You can do a pen and paper exercise, which allows you to figure out the space. And I can't show you that slide because there's a heavy duty slideshow crap here. Okay. Okay, let me go to another. Here, let's see. Yeah. You see this molecule here called Ba?
Yes.
That's called barbituric acid. It is different from uracil in the fact that it has this one extra hydroxyl group, right?
Yes.
You can basically use a pen and paper, literally draw out multiple 6-ringed and 9-ringed structures with exocyclic atoms which are capable of being a hydrogen donor or a hydrogen acceptor. So essentially nitrogen and oxygen containing entities. Yeah. You can do a pen and paper exercise and draw a lot of them. But systematically, if you wear a chemistry hat, which I am not very good at, but an organic chemist can look at these structures and say, not possible, not possible, not possible, and take a whole bunch of them out. Then there is a subset left, and then you can try and think of ways to try and synthesize them in the first place. And I'm happy to say some of these have been shown in prebiotically relevant reactions that have been carried out in labs. Some of these have been even showed in meteorites. These alternate molecules, right now your amino acids that you see in biology are 22 types of them.
Right.
But if you actually extract the Murchison meteorite that I talked about, right? And if you extract the organic aqueous, if you kind of take out some of the aqueous material that was drilled from the, if you try and analyze the stuff that went into the aqueous phase from the core, you actually see that other than the 22 amino acids that you have, there's a whole bunch of other amino acids which is not there in biology. So there is a notion that a process of molecular evolution, which happened over billions of years, which was shaped by the physicochemical constraints of the various eras, so to speak, eventually led to the selection of molecules that we see today.
Right. Okay. Fine. Yeah.
You veer away from this pH, your AUGCT are useless. They are happy at around pH 7.
Yes. Yeah.
Physiological pH. In some cases, they can tolerate up to pH 2, but they need special enzymes and so on and so forth, right, to protect them. But having said that, because you do have the same set of, they have other kinds of mechanisms, some of the extremophiles, to protect their internal biomolecules. But if you just take these biomolecules out of the cellular context and work with them at pH 7, they're happy. You veer away from pH 7, they will fall apart. That tells you that if you are thinking about where life might have emerged, under what pH, what temperatures, you will have to be open and think about other closely related molecules which might have been able to survive these conditions. And this is something that is happening in the field just in the last couple of decades. I will say less than a couple of decades. And we are one of the few people that think about what might this prebiotic landscape of bases look like. And this is a work that came out of Chaitanya's initial project, and it really kind of took off very well. And right now, one of the things which we want to do is to demonstrate this, which is, if you have, let me use this cartoon. If you have these sugar phosphate backbones, and if you have a bunch of nucleobases which look like cousins of your modern bases, which might much more readily form glycosidic linkages under harsher conditions, can you form informational molecules? That is something we are looking at right now in the lab.
Okay. I have one small question. Like,
sure.
So, like, we have heard about like organisms like tardigrades and all, which they are talking about that they could survive into cosmic space and all, and they could have traveled in meteoroids and from other planets and all. So, like, is that theory valid? How much of that is like?
So you're invoking to something called the panspermia theory.
Okay.
You know, I have, for me, by subscribing to the panspermia theory, I feel like we are changing the location of the problem from Earth to somewhere else.
Yes, yes.
That we can never understand. We do not have the technology to remotely try and figure that out. And let me tell you one thing. A lot of these exogenously retrieved material, I mean, material that keeps coming onto Earth, and it's been happening forever. There was a phase when it happened extremely with a very high frequency very early on in life's history, but even now it happens, right? When you actually drill these molecules and stuff, you can't see microorganisms in them. You see biomolecules in them.
Yes.
You see more primitive complex chemical molecules in them. When I say primitive, I mean they're simpler than the biomolecules, but you can see how they might have been the precursors for the biomolecules you're interested in. But nobody till date has shown any living entity in any of these. Your tardigrades, on the other hand, which you can readily hike up to the hill next to your house, pick up some soil from there, come back to the lab, and you can actually see tardigrades in them. These are entities that evolved on our planet. Based on the biochemistry we understand. There is also this, and tardigrades are known to survive a whole bunch of unbelievable harsh conditions, right? Unbelievably harsh conditions. That is because they can roll up and form this ball-like entities, and I'm forgetting the name of the process right now. It's just evading me, but the idea is that they produce something called trehalose, which essentially freezes all the biomolecules in their cells, in that it's almost like freezing in time. They go into this desiccated state where everything is frozen in time, and they will just stay like that forever. Give them water again, they open up and they start being tardigrades again. So for me, by saying panspermia is the way life came about on Earth, simply is transporting the problem to someplace else in the cosmos, which is extremely difficult to study or understand. For me, trying to understand what might have happened 4 billion years ago on Earth is a problem.
Yes. Yeah.
Because the geological, there's a whole bunch of debate on the geological settings. How reducing was the Earth's environment? Was it as reducing as in the Urey-Miller experiment? Or was it mildly reducing? There is still that debate, right? And you can only get some answers by looking at some of the geology, right? What are the molecules that might have been present in the prebiotic inventory? One can think about that when you understand the geology, and then you think about the aqueous rock interactions and try and understand what are the plethora of fundamental ions that can be possible in the space that can, and the different ways they can interact to form, or the other kinds of atoms that might be present that can interact to form molecules, and how they might react to the various ions present. So everything becomes more tangible if you want to try and understand how this process happened on Earth. Right. By transporting this problem elsewhere, I feel like you're just kind of taking the easy way out. And two, nobody has to date shown any living entity in any of these cosmic objects. Maybe if at some point in time in my lifetime, if it happens, I will be completely open to that possibility. At this point, I'm absolutely not open to that possibility. I don't know if you know about this one thing about tardigrades. Huh? Israel sent a shuttle around the same time as the Vikram lander thing, which happened, right? And in India tried to send a lander, no?
Right, yes. Okay.
Around September 2019.
Wow, perfect. Exactly, right? About 1 year ago, Israel tried to send a shuttle. There is reason to believe they might have actually sent a tardigrade experiment. The shuttle didn't work out. Something happened, things went very wrong. What have we done by the end of it? We have probably contaminated a cosmic object. And now when someday in future, we will get some of our Indian astronauts to get some samples back. And if there are tardigrades in there, voila, panspermia. But what did we do? We sent tardigrades from Earth. So it's a very, very tricky kind of a thing. For me, panspermia, is not something I can wrap my head around. It's much more easy for me to think about, systematically think about how chemistry might have transitioned to biology in the early Earth.
Thank you, ma'am. Yeah.
Thanks, Aditya.
Ma'am?
Yeah, who's this?
Yeah, this is Akash, second year BSMS.
Haji.
So I wanted to know, how would you know when you're looking at problems in this field, how would you know if you've solved them? How would you judge between competing theories? Because you can't really reproduce those conditions like the entirety of the world at that time. You could probably reproduce them selectively bit by bit, and you would never know whether what you've come up with is actually what existed back then. So what does it mean to be the leading theory amongst multiple theories? How do you decide between theories? How do you know you're more correct or less correct? What are the standards in the field?
Very tricky question to answer. You know, there's no way to systematically know if what we're saying is probably the way things happen, right? It's very difficult to try. There are no fossils. We cannot trace molecular fossils, right? You can trace organismal fossils which have this bony, which were made up of bones and stuff, and then you can still walk back and say, the dinosaur looked like this, this plant had this shape, so on and so forth. But you can't walk back and say anything like that with molecular fossils, because they barely, I mean, you have, of course, zircon signatures that can tell you how far back a biological process might have left some kind of a signature that will allow us to tell whether a lifelike process might have been plausible. That is all you can do. You cannot tell how that process panned out, or what are the niches that might have supported the process, so on and so forth. So by the end of all of this, if a scientist is truly honest, I think it's super fun to do this, because one, you try to understand at least small pieces of the puzzle. And when you are looking at some of these small pieces, you really, really, really have to think about the context systematically and convince yourself that the fundamental understanding that you have on which you're building this hypothesis is sound. And then from there you take and you try and understand the process, and it tells you what it tells you, and you try to understand what might have happened based on what you see. By doing all of this, while doing all of this, we learn a lot of very interesting biochemistry, right? Because we work in that interface. I'm sure a lot of people who are working in other realms of these areas learn to understand fundamental aspects of some of the processes that they're looking at. Whether they exactly panned out like that 4 billion years ago is anybody. Bunch of us in the field can start with fundamental precursor molecules and systematically work our way through the various stages that I talked to you about, and show that from the individual building blocks, we can build to a place where you can show the emergence of protocells, and from there you can show that the protocells behave lifelike, which is they can grow, they can divide, and they can evolve, and they can respond to new conditions that you throw them at. That might be the closest that you can get to say that maybe it happened this way.
I see. So it's not really about answering questions, it's more about triggering
It's more
patterns
about
of
understanding what are the various ways in which it might have panned out.
So it's a mechanistic understanding.
Of course, a lot of what you end up understanding is the fundamental mechanisms that underlie many of these processes, right? But to be able to say, मेरा process ही था जिसने chemistry को transition किया biology में on earlier, I think that's something that none of us can claim. None of us can make that claim ever. Okay. That's my notion. I think you should talk to a bunch of other astrobiologists if you come across them in the future and ask them and see what answers they have. But I feel like you can never say that. Even, let's say, the question that we are interested in, to understand how the molecules of an RNA world came about, how did they come about from the prebiotic soup, right? Whatever we show, whatever we try and understand, it's a iterative process. We know that going from the prebiotic soup directly to an RNA world, not possible. You can't start from RNA monomers and directly go to an RNA world, because it's a very difficult problem. So then you rework your hypothesis and say, you probably had other informational molecules that can be made much more readily, which potentially are even more simpler, but then eventually they might have evolved to form molecules of an RNA world, or molecules of an RNA world got selected for under a different set of constraints, so on and so forth, right? So you can do those kinds of things, but to be able to say...
Does it make it... do you have this idea of a better candidate theory and a worse candidate theory, or do you just have many theories?
There are a few different theories. So RNA world hypothesis is what came forth in my talk, right? But there is a competing hypothesis called the metabolism first hypothesis. And what they say is, you're having so much difficulty making this RNA molecule. In order for you to even make this complex molecule, you need a minimal metabolic network that made even more fundamental molecules. So then it's a minimal metabolic network that came about in the first place that then led to evolution of life as we know and understand. So this is a very famous competing hypothesis. There's a bunch of resources that I can share with the Science Club people, and then they can share it with all of you. And there are these lovely videos from Stated Clearly. I don't know how many of you know about this YouTube channel called Stated Clearly. And the guy has done quite a bit of videos on RNA world hypothesis, emergence of life, so on and so forth, chemical evolution or molecular evolution. And more recently, very recently, has also done this video on metabolism first hypothesis that kind of will give you a sense of how these competing hypotheses work. And I can also share this beautiful BBC article on origins of life. It's a pop science article, but it covers a whole bunch of research in both these realms, which talk about the work that's been happening in this area where we try and understand how you might have made these transitions. So maybe that will also help.
But you won't be able to really say which of these two is better or worse in any way.
There's no better or worse. You know, some, like I already alluded to when I showed you the two niches, the two competing niches, right? The terrestrial geothermal pools and the hydrothermal vents on the ocean floor. Some of the biochemical reactions in its very simplest complexity can very readily happen in the hydrothermal space, in the hydrothermal vent space. You try and do it in the geothermal pool space, it won't happen, and vice versa. So then the question is, where did what happen? For me, the only reason the hydrothermal vent hypothesis, as much as it's given a sort of, very interesting, there's a lot of interesting work coming out in the metabolism first realm from the hydrothermal vent space. I have a problem of trying to understand how we would work around the dilution issue, right? Let's say all these lovely black chimneys that you saw.
Yeah, okay, so they're just going into the ocean and dispersing
they're called black smokers. A lot of interesting chemistry can happen within the smokers. But as soon as you form these fundamental biomolecular entities, right, you're going to hit the ocean and then dilution. So the terrestrial geothermal pool is a much more constrained space. One can kind of keep track of the reactions that can potentially be happening at the edge where you have a lot of clay lining. So a lot of interesting surface chemistry is possible. Yes, of course, the hot spring might be very deep down there and we don't know what's happening down there, right? But having said that, a lot of interesting surface chemistry is possible where molecules get constrained on various surfaces and so they're not floating away into in directions where they'll never interact with each other. So it's more tangible for me as a niche. Hydrothermal vents, very cool in terms of some of the chemistries they can sustain, but to imagine if all of it happened there and then how do you deal with the dilution problem? I can't wrap my head around that.
One more question, like totally different thing though. If I look at RNA, as you said, it has a very high mutation rate. So can things like viroids and by extension RNA viruses, is it a forlorn hope to think that they might tell us something about the RNA world? Because despite being RNA entities, they'll have evolved a lot since. Or is it possible to extract some information about what the RNA world may have looked like?
No, I mean, if you do think about some of these RNA viruses, right? Of course, some are very complex, but some of the more simpler RNA viruses literally have a nucleic acid and they have some kind of a small protein coat and maybe a little bit of lipid, but even then that also might not exist. Maybe some of these entities, I can't.
Viroids don't even have the coat, so that's
the viroids don't even have the codes, exactly. So maybe they can at least tell you the kind of interactions, biomolecular interactions that are crucial even for their emergence in the first place. So those are the kinds of things one can glean from studying some of these things. If you go to more complex RNA viruses, they even have the SARS-CoV-2 has a lipid coat, if I'm not wrong, right?
Yeah, it is. It's a membrane-bound virus.
Exactly. So one can even try to think about some of these. Your protocells might have possibly looked like much more primitive versions of these SARS-CoV-2 viruses, where you already have 3 different biomolecules interacting, right? Your lipids, your amino acid-based biomolecule, and your nucleotide-based biomolecule. All 3 are already present here.
But the mechanism by which the lipid is sourced is one that depends on cells existing. So that couldn't possibly be relevant.
Exactly. And hence, that's the reason I didn't, when I said the definition of life, I didn't put viruses there because viruses are obligate parasites. Yeah, they are alive only in the context of another living entity. Take the living entity out of the picture and I don't know.
So the question is really that if you, if you think about the virus, then the question is whether it could have preserved anything from that period. So on the one hand, you have reverse transcriptase playing a crucial role in group 6 and group 7 viruses, retro and pararetroviruses. You have you have just RNA based stuff in viroids,
Right.
but on the other hand, again, there's no guarantee that they resemble what was there early on. They may have evolved completely. So
Independently.
I just don't know whether they're probed as a source of information about the RNA world.
So there are
And also,
very few people who take this approach that you're talking about, right? A good number of us take a bottom-up approach because we think that this particular space is complex enough that we probably, I don't know. I'm trying to think. I do not know of too many colleagues in the field who are looking at this at all. There's this one person that I recently learned about. He's in Australia, and he kind of talked about how there's a fourth domain in life, and that is this big, what did he call it? If I can find my notes, I'll tell you. He called it Viria or something like that. And he says that when you look at viruses, you realize that they deserve their own domain because of all the amazing complexity that is involved in the space. So,
I
but
think right now there's a lot of debate about whether viruses arose once or multiple times over. So if you're going with a cladistic approach, you couldn't even call it a single domain. So the problems
right.
there.
There is that too. Exactly. So I mean, his talk led to a lot of discussion later on. But yeah, I had never heard of this Viria or whatever he called it prior to his talk ever. So it looks like there are people who think about these things, but I haven't seen too many of the prebiotic chemists looking at the space to try and see if you can learn something about the RNA world from that angle. We in our lab are very interested in the tripartite crosstalk between membranes, nucleic acids, and amino acids, because to imagine the RNA world in isolation would be a little naive. And then your prebiotic soup is super heterogeneous. There was no reason to believe that it was only made up of mononucleotides. If you actually look at the Urey-Miller experiment, your amino acids form much more readily. You don't see nucleotides or nucleosides in them. So then to completely disregard this other class of molecule by focusing on RNA world and saying that RNA world was how it happened, and then you had to evolve a ribozyme in that RNA world was essentially came about, which then could stitch up amino acids, and only then you had proteins. I feel like it's a tad naive. It's much more difficult to abiotically form peptides, but increasingly a few of us in the field are showing that you can do it much more readily. And Manish, one of the grad students in the lab, is very interested in this. And yeah,
Could
there
you have,
should be this.
could you have proteins carrying information? I mean, I know that doesn't happen in any. Could you have proteins carrying information?
That's the problem. So there are two ways to thinking about information. In proteins, the information that is encoded for or in complex protein assemblies is structural.
Yeah.
Like in prions, right? You can actually replicate the fundamental scaffold. But again, this is like something that not many, information in a protein is non-trivial. So maybe there was a kind of a molecule which combined features of both the peptides and the nucleic acids, and maybe one of these proto-RNAs could have been one of these primitive molecules which had some of the characteristics of both your amino acids and your...
I mean, RNAs can also carry structural information, right? RNAs, when they act in a catalytic way, they do form...
Of course, they do. But they can also encode information as we understand information transfer, right?
Yeah.
Because fundamentally at the level of being able to make these hydrogen bonds with complementary bases. In your proteins, you don't have that kind of an information encoding.
Wait, so do you envisage this as being one RNA carrying information, producing another RNA that then acts on it, or are they just or are the RNAs doing the catalytic work and the RNAs being transferred?
We don't even need a ribozyme to be able to replicate information. In our labs, we do it completely non-enzymatically. Minus the ribozyme also. You just need the template, you need the primer, and you need the nucleotide. You just tinker with a little bit of chemistry and you can replicate information in the template strand and make a full complement of the template strand, completely without enzymes. I'll be happy to talk about that on the side.
Very cool.
But thank you all for staying back and for this fun interaction. And any one of you have any other questions, please hit me up and I will share a bunch of resources with Science Club. And can you please forward that, Krishna, to people who are interested? Maybe not everybody wants it, but if somebody, I think you can still send it out to all the email addresses that you sent it out to, and whoever wants to read up can read up.
Yes, ma'am.
Yeah, sounds good. Thank you all for your time. Ciao.
Thank you for this amazing topic.
Thank you, Krishna. Thanks. I'm sorry if I fumbled somewhere. Chalo, bye.